Hello everyone. Welcome to the new session of Plant Kingdom. This is the fourth part of this chapter, and in this part we are going to study about the pteridophytes. So pteridophytes are the seedless plants. They are vascular or cryptogamic plants. So first time the vascular structures appear in this particular type of plants, that is called as pteridophytes. Sporophytic plant body. Now here comes the sporophytic plant body. In the earlier class for the bryophytes, we are saying or we are considering as a gametophytic plant body. But here in the case of pteridophytes, we are calling it as a sporophytic plant body. So very, very important. This is the main difference between the bryophytes and pteridophytes. Inconspicuous gametophyte. Now, inconspicuous gametophyte means gametophyte is comparatively for a very short duration of time. And if we recall to the bryophytes, there the gametophyte there the sporophyte was inconspicuous. They contain small sessile antheridia. Sessile antheridia means no stalk-like structure is present. Okay. So no stalk-like structure. Antheridial structure is like this. Like this. So no stalk-like structure. And it is partly embedded in the thallus-like structure, whereas archegonia with 4 rods of neck. Now this is first time appearing the archegonia surrounded with the 4 rods of neck. So this is one of the very important characteristic of the pteridophytes. Vascular tissue developed for the first time in pteridophytes. So this is the first time that in plants vascular tissue can be seen. So that is why they are also called as vascular cryptogamic plant. Roots. Adventitious roots arise from the rhizome. Now here the stem is flattened like structure. It is not vertical structure, it is a flattened horizontal structure from which leaf originates. Okay, from this leaf originate, and roots also originate from this rhizome like structure. The stem is in the form of rhizome. So this is a rhizome structure. Leaves are pinnately compound. Pinnately compound means if this is a leaf, then there are pinna-like structure. So this is a compound leaf. I'm just drawing a one leaf to make it clear that what is a pinnately compound leaf. So this is a pinnately compound leaf. So this is a compound leaf structure. Leaves may be small as seen in Selaginella. So sometimes leaves are very small as in the case of Selaginella. They are known as microphyls. So they are very small in structure. That is why they are called as microphyls. Larger leaves are also known as macrophyls. When the leaves are larger, they are called as macrophyls. Ramenta. Now ramenta are the hair-like structure. Now here you can see this is a young leaf. A young leaf is a curl-like. It is a curled young leaf. And on this, hair-like structures are present, which is called as ramenta. Okay, so they are the specialized hair-like structure found on the underside of the leaves or fronds in ferns. So in case of ferns, they are present on the underside of the leaves or fronds. So they serve as various functions, including protection. They provide some sort of protection. Also helps in reproduction and water conservation. Circinate vernation. Now, this is also one of the very important characteristic of this particular pteridophytes. Circinate vernation. So, it refers to the specific type of a leaf or a frond arrangement that is commonly found in the pteridophytes, particularly in ferns. So, when the leaves in the ferns are new, they originate in like this. It is curled. New leaf is curled. So, here you can see this structure. The new leaf is having curled in shape. So young leaves or the fronds of the fern gradually uncurl or unfurl from the tightly coiled or the rolled up position as they develop. So when they develop, they uncurl it and they expose its leaflets. Okay. So this is circinate vernation. So sometime in the examination, it can be asked what is circinate vernation or in which type of plants it is found. In pteridophytes, reproduction do not occur by seeds. Okay, no seeds. Instead, by the spores. So, seed-like structures do not form in the case of pteridophytes, whereas spores are formed. So, where they are formed, where they are present. So, spores are formed in a special structure called sporangia. Now, where this sporangia is present. So, if you take a fern plant and see the ventral part of the leaflet. Now, I'm just drawing one leaflet and here you can see some circular brown structures, reddish brown structures like this. So these are called as this whole leaf-like structure which is bearing sporangia are called as sporophylls. Phylls means leaf-like structure and sporo means bearing spores. So these are the leaves which are bearing spores in it. Okay. And these spores are present where? In sporangia. Where this sporangia is placed, it is present in a ventral surface of the leaf structure. That is why it is called as sporophylls. Now, the sporangia are typically located on the underside of the specialized leaves called sporophylls. Now, sporangia are reproductive structures found in pteridophytes. So, here you can see one of the sporangia. This is the sporangia. And here, when it get ruptured, this wall get ruptured, all the spores present inside it comes out. And when these spores get a suitable substratum having water, suitable temperature, okay, they germinate. So inside the sporangium, specialized cells undergo meiosis. Not all cells, specialized cells undergo meiosis to produce haploid spores. Spores in majority of the pteridophytes are similar. All are same types. So that is why they are called as homosporous. Okay, homosporous. So examples of the homosporous are Pteris and Adiantum. These are the two ferns. Okay, and they just bear single type of spore. That is why they are called as homosporous. Now, there are some pteridophytes that give rise to two types of spores. If you see the size of spores, some are very small in size. That is why they are called as microspores. And some are comparatively larger in size. They are called as macrospores. Microspores give rise to male gametophyte. Now, when these microspores are released at a suitable temperature, they get water, they get germinated and form a thallus-like structure. So, this gives rise to a new plant which is called as male gametophyte. And megaspore give rise to the female gametophyte. Okay, so the plant which bear two different types of spores, that is the microspores and macrospores. The examples are Selaginella, Salvinia, and Marsilia. These are the three very important examples are heterospores, means the plant which bear two different types of spores. Okay, now this is also one of the very important characteristics, strobili or cones. So now you have seen in the case of fern that leaf bear sporangia, and inside the sporangia spores are present. Okay, so leaf-like structure having sporangia, they are called as sporophylls. Now in some cases, these sporophylls, some cases means in some plants, sporophylls may form distinct compact structures. Okay, they become very compact structures. All the sporophylls unite together and they form strobili or cones-like structure. Okay, strobili or cones-like structure. And examples of the pteridophyte which are having strobili or cones are Selaginella and Equisetum. So this is also very important as from examination point of view. Seed habit in pteridophyte. The first time seed habit can be seen in the plant, this plant that is pteridophytes. Selaginella show traits which resemble the characteristic for the formation of seed. Okay, so they are saying that Selaginella is having some traits, means if see the evolutionary point of view, then we can see or we can resemble it that this is the starting of formation of seed. What are those? The megaspores and microspores germinate and give rise to the female and male gametophytes. So two different types of spores are there, megaspores and microspores. So the female gametophyte in these plants are retained on the parent sporophyte. Okay, so from the microspores, the spores or the sperms are released from the parent body. But what about the female gametophyte, that is the megaspore, that remain attached to the parent sporophyte for the variable periods. Okay, so this is showing that it is having a seed habit. The development of the zygote into a young embryos take place within the female gametophyte only. So after the fertilization, the zygote is formed. That zygote is formed in the parent body only. Young embryo is also formed. That is also attached to the parent body only. Okay. This event is the precursor to the seed habit. Now this is the showing that in this way, the seed habit has been started and it can be considered an important step in the evolution and formation of the seeds. So, to have a clear-cut difference between the homosporous and heterosporous, let us see this table. If we talk about the homosporous, all the sporangia are similar. Okay, sporangia are similar. Why? Because they bear same types of spores. Here the sporangia are two types, two different types of sporangia. One which bears the larger spores, that is called as macrospores, and one which bears smaller size spores, that is called as microsporangia. Spores are of one type only. Spores are of two types. Larger, that is called as megaspore. Smaller, that is called as microspores. Gametophyte is one type. So only one type is spore is there. So that give rise to one type of gametophytic plant body. But here in the case heterosporous, they are having two different types of spores, microspores and megaspores. So they give rise to two types of gametophytic structure. One is male and another one is female. It has no evolutionary significance, but heterosporous is having evolutionary significance that gives the seed habit. Okay, so this is also one of the very important characteristic, that is first time this structure appears, that is the prothallus. So prothallus developed from the haploid spore. So when the spore germinate, okay, it germinate and form a thallus like structure, and later it get converted into a heart shaped structure like this, a heart-shaped structure, dorsiventral flattened. So dorsal surface of the prothallus, lower surface of the prothallus. The lower surface of the prothallus bear rhizoids. Now here you can see hair-like structures are present, which is called as rhizoids. Prothalli produced specialized structures called gametangia. This is very important. Gametangia are present on the lower surface or the ventral surface of the thallus. Antheridia, these are the male gametangia that produced and release sperm. So if again I draw the thallus like structure like this, okay, and here are the rhizoids like structure. Here some antheridia are present, okay, that give rise to sperms. And archegonia at this particular stage or at this particular part, female structures are present, okay, which is called as archegonia. Photosynthetic tissue. So upper surface of the prothallus contains cells that capable of photosynthesis because they are having chlorophyll. They are having chloroplast, and inside the chloroplast, chlorophyll is present. So they can perform the process of photosynthetic tissue. So this particular part is photosynthetic tissue, and they can perform the process of photosynthesis. So let us classify the pteridophytes. Pteridophytes are like bryophytes, are three different types. Pteridophytes can be divided into four different types. The number one is Psilopsida. Psilopsida. Most plants are fossil. For example, Xylotum. Mostly the individuals are not present today. They are present in a fossil form only. So most of the members of this particular plant are fossil. Second one is Lycopsida, commonly called as a club moss or the spike moss. So this example is Lycopodium and Selaginella. Sphenopsida, they are called as tree ferns. They are quite larger, and its example is Equisetum. Phelicopsida, they possesses fan-like leaves. Now here you can see leaf are fan-like structures. So these are, they possesses fan-like leaves. Example is Marsilia. So four different types of pteridophytes, namely Psilopsida, Lycopsida, Sphenopsida and Phelicopsida. Okay, let us have a overlook at the life cycle of pteridophytes. So this is the main plant body, which is called as fern plant. This is an example of a fern. Now here you can see it is a mature sporophyte. Okay, and at the ventral surface they are having sporangium. They are having, this is one leaflet structure showing. This is the sporangium, and this is showing one sporangium. Now inside the sporangium, this is diploid structure. This is diploid structure. This sporangium is also diploid, but inside the sporangium there occur a reduction division which is called as meiosis. Okay, so inside it spore is formed which is haploid structure, and I'm showing it with the N, haploid structure. Here also you can see this orange part, this is sporophytic structure, and this green part is showing the haploid structure. So spores, they release, they germinate and give a prothallus structure. This is prothallus structure. Now this prothallus is having both antheridia and archegonia. So here you can see from antheridia sperms are released, and here in the archegonia egg is present. So when one of the sperm reaches to the egg, it fertilizes the egg and it forms zygote structure. Now this zygote germinate and give rise to a sporophytic structure. So let us see life cycle of pteridophytes. The life cycle of pteridophytes alternate between two main phases, that is the sporophytic stage and gametophytic phase. Sporophytic phase is the dominant phase, as we have seen in the case of bryophytes. The gametophyte was the dominant one, but here in the case of pteridophytes, sporophytic phase is the dominant in the life cycle. The leaf or the frond bear plant, also known as ferns. Within the sporangia, spore mother cell undergo meiosis to produce haploid spores. We have seen that inside the sporangia, spore mother cells, special cells that undergo reduction division or the meiosis and produces spores. Okay, after the formation of spores by the reduction division, those spores are released from the sporangia, and at the suitable substratum they germinate and form a heart-shaped prothallus-like structure. Prothallus-like structure. It is a small heart-shaped gametophyte structure that develops from the germinated spore. Gametangia formation. Now, on this prothallus, only specialized structures called gametangia develop. Gametangia means reproductive structures, that is the male reproductive structures and female reproductive structures. Antheridia and archegonia. The antheridia produce sperm cells and archegonia produces egg cells. Okay, so this male part, that is called as antheridia, it produces sperm cells, and the female part, that is called as archegonia, it produces single egg cells. So after the fertilization, now what happened? For the process of fertilization, the sperms which is released from the antheridia, it swims as the water is available and it reaches to the egg. It fertilizes egg and forms the diploid zygote. The diploid zygote is formed. The new sporophyte plant grows from the prothallus. Okay, now from this prothallus, a new plant grow as a sporophyte. Okay, so let us talk about the importance of pteridophytes. They are very important. They are economically very important plant. How? Some of the pteridophytes are ornamental in nature. So ferns are popular ornamental plants and they are planted in garden, parks and indoor spaces because of their beautiful look. Medicinal uses. Some pteridophytes have been used in the traditional medicine for the potential medicinal properties. So some of the pteridophytes do have medicinal properties. That is why they are used in making medicines even. They also helps to control the erosion Okay, so some ferns are used for the erosion control on the slopes and in the areas prone to soil erosion. So we have seen bryophytes are also helping in lowering the erosion, and pteridophytes are also helping in the erosion control. Fodder and forage. Some ferns are used as a fodder and forage. So they are used by the cattle. Ecological importance. They provide habitat and food source for the range of animals. Scouring. Now this is very important. Equisetum plants is used in very different way. Equisetum stem have been used in scouring, clearing of utensils and polishing of metals. This is very important. Nitrogen fixation. Azolla as a symbiotic association with the nitrogen fixation cyanobacteria Anabina azole. Now this is cyanobacteria which resides in the body of Azolla, which is the Pteridophytes, and help in nitrogen fixation. So this is all about Pteridophytes. We will meet in the new session and talk about gymnosperms. Thank you very much.